Hello, my name is Catarina. I am from I3S, University of Porto, and we are part of the GENEGUT project. So today I'm here to explain you what is a 3D cellular model and how does it work. So in our lab, we have been investing in the development of these 3D models to study Crohn's disease and to address the potential of using this model to screen novel therapies. And what are these models? So if you think about an organ, these are not 2D flat structures. These are 3D structures with specific architecture. So in our lab, we have been reassembling this type of structures, in particularly for the intestine, with different layers of cells, resembling the native structure of the intestine. At our lab, we want to mimic the intestine to study Crohn's disease, and for that, we have been developing a mini-intestine on a lab dish. And basically, how does this work? We set the culture of different cell types and a 3D structure that will mimic the different layers of the intestine and also its particular architecture. And this is important because it will resemble in a more biological way the tissue that we find in the intestine. And to resemble also Crohn's disease, we can treat this tissue in the lab with the different molecules that will mimic the inflammation that occurs during the process of Crohn's disease. And why is this important? Because during GENEGUT, we are developing a different set of nanoparticles to treat orally Crohn's disease. And these models will be relevant to test in an in vitro setting the efficacy of these therapeutics before moving forward for animal or even to human clinical trials. At this stage, our models are already developed and they will be used to screen the best candidates developed under this project to move forward to animal studies.